Hello everyone. In this video we are going to talk about metabolism. Have a look at this diagram. Can you guess what this is about? You can pause the video and think about it. Okay. There is a substrate, there is an enzyme, and that would be the combination of the substrate and enzyme over here. That's the way it starts. And then you will see enzyme number 1, and then enzyme number 2, and then enzyme number 3, and then finally that would be an end product. And this end product will back to the first enzyme, and it attached to some area of the first enzyme. And you see that something happens over here after the attachment of the end product over to the first enzyme, and this location will change its formation. See, this is the previous formation is like this, but this formation is like this. So this is something as an example that you are going to learn by the end of this section, so don't worry about if you don't know what's going on in this diagram. Let's see what is the essential idea. Metabolic reactions are regulated in response to the cell's needs. So we are going to completely understand what are the metabolic pathways and metabolic reactions. However, most of the part of the unit 8.1 have been already discussed in our previous units, which was in unit number 1. Well, this is an explanation regarding to inhibitor effect, and it is about the enzyme which is having the mechanism for inhibition, and this is the inhibition process. This is this is what I just explained. I'm not going to go with details because this would be a very a part of the unit that we are explaining later. Okay. So what we are going to understand and what are the application and the skills, we will learn about metabolic pathways, which consist of chains and cycles of enzyme catalyzed reactions. And then enzymes lower the activation energy, the function of the enzyme, by lowering the activation energy of the chemical reactions that they catalyze. Enzyme inhibitors can be competitive or non-competitive. This is also something that we discuss. And the inhibition process. Metabolic pathways can be controlled by end product. This is what I just showed you at the first slide, how the end product can control the metabolic pathway. And end product inhibition of the pathway that converts threonine to isoleucine. We will learn one example regarding to this inhibition product. Use of databases to identify potential new anti-malarial drugs, and calculating the plotting rates of reaction from raw experimental results, and distinguishing different types of inhibition from graphs and specified substrate concentration. So these are the things that we are going to learn by the end of this unit. So let me know how much you can guess from this word until reaching to this word. So you can put some other letters and then finally get the last word as blink. This is a kind of example of how an enzyme from the first step is becoming to the enzyme in the last step after like 2, 3, 4, 5, 6 would be the 7, and there will be like additional 5 steps in between in order to reach to the final product. So this is the challenge. By changing just one letter at a time, get from thread to blink. All intermediates must be real English words. This is a challenge. You're going to stop pause the video and try to find the words. Okay, did you get it correct? So you could make by changing just one letter by letter, like T become B, like bread, and then breed, and then R to L, bleed, and then blend, and then blind. Everyone who could do it correctly, you're amazing. I didn't think that was an easy job. Okay, so metabolism, the sum total of all chemical reactions that occur within an organism. And what do we call as metabolic pathway? All the cycles or chains of enzyme catalyzed reaction, those are all called metabolic pathways. And the chemical change from one molecule to another often does not happen, not in one large jump. This means that it doesn't happen it is not the way that thread is becoming blink in one jump. So it will be step by step, okay, one letter at a time, but in a sequence of small steps. The small steps together form what is called a metabolic pathway. Okay, let's watch this SWF file. If you want to watch it by yourself after having the PowerPoint file, you need to download this SWF file.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Metabolic pathways could be cycles or chains, which are catalyzed by enzyme. We have two examples, and both of them can be observed. One of them is glycolysis, which is a part of respiration. And in glycolysis, glucose will be converted at the end of this chain to pyruvate, and it is an example of a metabolic chain. And glucose, by the usage of 2 ATP at the beginning, it converts to a molecule which is hexose biphosphate, and then with this hexose is becoming 2 triose, and these 2 triose biphosphate can be converted into 2 pyruvate at the end, and the production would be like 4 ATP and 2 NADH. We are not discussing here the details in glycolysis. What we need to know over here is just that glycolysis is a part of a metabolic pathway as a form of chain, metabolic chain. And Calvin cycle, which is a part of photosynthesis, not respiration, it is an example of metabolic cycle because it is like a cycle. So the carbon dioxide will be fixed, which means that will enter to the cycle by adding to ribulose biphosphate, and by the Rubisco enzyme it becomes a molecule which has 4 carbon, and by the usage of the ATP and then converting to other molecules, it becomes it continues with the cycle to become a 4 carbon molecule, and then to become ribulose biphosphate, and by the usage of the ATP here. So again, we are not discussing what happens to the Calvin cycle, which is the end product of the Calvin cycle is sugar, because that's what is happening during the photosynthesis in light-independent reactions, production of the sugar by the usage of the energy which has been already produced in the light-dependent reactions, and by the entrance of the carbon dioxide. So that's what happens in the Calvin cycle and in photosynthesis. But anyway, the discussion over here is this is a metabolic cycle and this is a metabolic chain. Activation energy. This is a very nice topic. We have discussed this before, and you just need to look at the graph and you will see that with the presence of the enzyme, that would be so much difference with the usage of the energy, or the first step, which is activation energy, will be lower down if there is an enzyme. So imagine that over here, which is the energy required for activation of the reaction. With the presence of the enzyme, this energy level would be at this level. But if there is no enzyme, then the energy level, the activation energy, will be higher. So which means that we need to have more energy in order to activate that reaction, make it possible to be done. And that's one of the functions of the enzymes, to lower the activation energy. Okay, so the initial input of energy that is required to trigger a chemical reaction, that is called activation energy. And enzymes benefit organisms by speeding up the rate at which reaction occur. They make them happen millions of times faster. How do enzymes lower the activation energy of a reaction? So we can go step by step. First of all, the enzyme here will match to substrate, and there is an active site. So there will be the enzyme substrate combination. And then next step would be enzyme substrate complex, and enzyme changes shape slightly as substrate enters active site, making the fit more precise. So if there is a little, you can see that if there is a little different in active site, enzyme try to fit it in order to exactly fit with the substrate. And then the enzyme substrate complex will be formed. And the next step will be the enzyme product complex, which means that the enzyme will do whatever it needs to do, and this substrate, it's not already the first substrate, it has been already changed to a product. And then the last step is that those products will be released, so enzyme product leaving active sites. The substrates bind to enzyme active site, and the active site is altered to reach the transition state, and then due to the binding, the bonds in the substrate molecules stressed or become less stable, and the binding lowers the overall energy level of the transition state. So this binding over here and this complex lower the energy required without the presence of the enzyme, which means that this is the reason that enzyme can affect. The activation energy of the reaction is therefore reduced. And then nota bene, which means note well, the net amount of energy released by the reaction is unchanged. There is a website and a link over here which is talking about enzyme inhibition. So from now on, that would be the enzyme inhibition examples and explanation. Since we have a lot of slides related to this enzyme inhibition, I'm going to explain them, and then I will go back and play the video and watch the video together.